All right, well, welcome everybody. Very happy to see such a big group here. My name is Will Prince. I'm at UT Southwestern, and I'm very pleased to be introducing Mike Skowra today. Mike is really growing into a leader in organelle biogenesis and membrane rearrangements and membrane biology. So Mike got his bachelor's degree from St. Louis University, and then he went on to do a PhD in Phyllis Hanson's lab at Washington University while she was still there. And he did some really groundbreaking work on escort proteins, particularly discovering a role for them in membrane repair. And after graduating, he went on to Tom Rapport's lab, where he's been since 2018. And again, Mike has done really groundbreaking work, sort of reviving the sort of moribund field of peroxisome import, and really made some really seminal discoveries again on understanding the import cycle mediated by PEX5, which is a key import protein. And then more recently, publishing really an amazing paper that reveals the long, mysterious mechanism by which peroxisomes import proteins. And so with that, I give it over to Mike, and I'm pleased to have you here. Thanks, Mike.
Great. Well, thank you, Will, for that kind introduction. And also to Yihong, thank you for the invitation to speak at this forum. It's my pleasure to attend. I'm hearing some background noise from someone. I'm not sure what that is. Okay, so I thought I would start by first drawing your attention to this iconic electron micrograph in the background here. So this image comes from George Pallotti. It shows a cross-section through the cytoplasm of a rat liver cell. And it showcases many of the structures that we commonly associate with a eukaryotic cell. So for example, you can see mitochondria with their characteristic cristae along the top of the image here. There is also the ribosome-studded endoplasmic reticulum, or ER, which shows up as these long tangles, and also as cross-sections through ER tubules in the middle here. And then in the bottom left corner, you can see peroxisomes. And so they appear as these round bubbles that contain these characteristic electron-dense inclusions shown here in black. And so peroxisomes, like the ER in mitochondria, are of course organelles that are found essentially in all eukaryotic cells and organisms. And like Pallotti's image nicely illustrates, that peroxisomes are mostly spherical, and they are enclosed by a single lipid bilayer. And they are also quite small, often being under a micron in size. The predominant function of peroxisomes is to break down a variety of different substances and biomolecules by oxidation. And so these can include specific types of amino acids and nucleotides, also many toxins and drugs, and in particular, fatty acids, which in many eukaryotes can only be broken down inside peroxisomes. Peroxisomes can also play biosynthetic functions. So in humans, arguably the most important among these is the production of essential precursors of bile salts in the liver, which are required to emulsify fats in our diet, and also the production of precursors of ether lipids, which are a major constituent of the myelin that enchefs our neurons. And as I hope you can appreciate from these diverse functions, peroxisomes are actually vital for human health. And mutations in peroxisomes that compromise their functions cause a spectrum of devastating disorders in humans. And arguably the most tragic among these is Zellweger syndrome, which can result either from the complete absence of peroxisomes or the inability to import proteins into the organelle, and is therefore almost always fatal early in life. So like most organelles, peroxisomes cannot make their own proteins. These are encoded by nuclear genes, and they must first be synthesized in the cytosol and then transported across the organelle's limiting membrane into the peroxisomal lumen. This is also called the matrix in the field. Now, in the case of most organelles, like ER in mitochondria, proteins can only be imported as monomers in an unfolded conformation. This is in large part to ensure that a tight seal is maintained around the polypeptide as it is moving across the lipid bilayer. But peroxisomes, surprisingly, are a bit different. They have long been known to be able to import proteins after they have already folded in the cytosol, or even after they have formed oligomers or assembled into complexes without disrupting their integrity. Now, conceptually, this, of course, vaguely resembles nuclear transport, but the underlying mechanism has been a long-standing mystery in the field. I'll also point out that import into peroxisomes is remarkably efficient. The proteins are essentially pumped into the organelle against the concentration gradient to the point where some of the imported proteins even crystallize out inside the organelle and form these electron-dense inclusions that are characteristic of peroxisomes in many electron micrographs. And so we've been very interested to understand how peroxisomes can import folded and hydrophilic proteins that have very different geometries and different surface properties against the concentration gradient and also across the hydrophobic limiting membrane of the organelle. So the basic principles of the underlying pathway have been worked out by seminal work from many groups over the past several decades. And it's impossible to acknowledge everyone's contribution. So the best I can do is to encourage you all to consult any number of the great reviews on this topic, which are easily accessible in the literature. So peroxisomal proteins often contain a peroxisome targeting signal, or PTS. And among these, or in most cases, the signal is called a PTS1, and it consists of the amino acids serine, lysine, and leucine, or variants of it, at the very C-terminus of the protein. And the signal is also often connected by a flexible linker to the remainder of the peroxisomal protein, as shown here in this crystal structure of a human peroxisomal enzyme. So peroxisomal proteins are then recognized in the cytosol by protein factors called import receptors. So these are all highly homologous to each other, but arguably the most conserved and the most important is a receptor called PEX5. So PEX5 consists of a long, unstructured N-terminal region, which is characterized by two features: a conserved cysteine at the very N-terminus, and also a number of these short aromatic motifs, shown here in blue, that are characterized by the amino acids tryptophan and also phenylalanine or tyrosine. And this unstructured region is followed by a number of tetratricopeptide repeats, or TPRs, at the C-terminus. And the TPRs fold into a globular helical bundle, which directly binds to the PTS1 peptide on many peroxisomal proteins. So the import receptors, like PEX5, play a central role in the underlying import pathway. They recognize the peroxisomal cargo in the cytosol, as I've just told you, and then they facilitate the translocation of the bound cargo across the peroxisomal membrane into the peroxisomal matrix. And of course, how this translocation step actually occurs has been a long-standing question. Now, it turns out that PEX5 and the other receptors don't act alone. They have to cooperate during import with two small membrane proteins on peroxisomes called PEX13 and PEX14. Historically, PEX14 has been the subject of much attention, in large part because it is well known to bind very strongly to the import receptors. But between these two proteins, it is only PEX13 that is absolutely essential for import to occur in vivo. And this, of course, argues that PEX13 very likely plays a more fundamental role in this process. And so what is PEX13? So PEX13 is a small protein. It consists of only two conserved features, both of which are required for import to occur. The first is a very long amphipathic helix, or AH segment, often over some of the amino acids, if not longer. And the second is an unstructured domain called the YG domain. In some species, PEX13 also contains a transmembrane segment, or TM, and other domains, although these are much less conserved. So the YG domain in particular is characterized by a repeating pattern of aromatic amino acids, predominantly tyrosines, which are shown here in pink. And these are connected by short flexible linkers composed mainly of serine and glycine. And I hope you can appreciate that these sequence properties are more or less conserved, even though the actual amino acid sequence of the YG domain varies quite a bit between eukaryotic organisms. The sequence properties of the YG domain also uncannily resemble those of FG nucleoporins. So FG nucleoporins are proteins that also contain large unstructured domains, except in this case, these domains are characterized by repeating phenylalanine residues connected by short flexible linkers. So very similar to the YG domain of PEX13. And based on the work of Dirk Gerlich and others, FG nucleoporins are thought to form a dense meshwork within the central channel of nuclear pores. And this meshwork, it resembles a hydrogel and appears to be held together by cohesive interactions between the phenylalanine residues in the proteins themselves. And the meshwork normally functions to prevent most proteins and other large molecules from moving across nuclear pores. However, nuclear transport receptors, or NTRs, can selectively partition into this hydrogel phase, and this allows the NTRs to shuttle a variety of bound cargo, especially folded proteins and protein complexes, across nuclear pores. And so the analogy to the FG nucleoporins raised to us the very provocative idea that perhaps the YG domain of PEX13 might play a similar role on peroxisomes. So to test this idea, I tried purifying the YG domain of PEX13 and asked whether it would form a hydrogel in vitro. And the answer is, of course, yes. So a solution of the recombinant purified YG domain very readily forms hydrogels in solution. And these essentially retain the shape of the container in which they were originally stored. Now, interestingly, when the tyrosines in this domain are mutated to serines, then the resulting fragment remains completely soluble and no longer forms a hydrogel. Even at the same concentrations where the wild type readily forms gels. And so the easiest interpretation of this is that the hydrogels are held together by cohesive interactions between the tyrosine residues. So analogously to how the FG meshwork in nuclear pores is thought to be assembled. Now, if the YG hydrogel functioned as a selective phase, you would expect that peroxisomal import receptors should partition into this material and accumulate inside, whereas other proteins should be excluded. And to test this hypothesis, I devised an assay whereby I prepare small drops of the YG hydrogel in a glass bottom dish. I bathe them in buffer containing different fluorescent proteins, and then use microscopy to monitor whether the proteins enter the hydrogel or stay behind in the buffer. And what I find is that indeed, the peroxisomal import receptor PEX5 fused to green fluorescent protein, or GFP, very readily enters the YG hydrogel and becomes greatly enriched inside the hydrogel over time, as shown here in these static images. And then if we measure the concentration of the PEX5 GFP in these images from left to right, then I hope you can appreciate that the receptor, again, reaches concentrations in the hydrogel many times greater than the concentration of the receptor in the buffer. And in addition, I also observed this characteristic depletion of receptor molecules in the buffer just outside the gel edge. And we interpret this depletion to mean that the receptors partition into the hydrogel so quickly that the diffusion of non-partition receptor molecules in the buffer cannot keep up with it. And now contrast that to the behavior of GFP alone, which shows no accumulation in the YG hydrogel at any time point, and also no characteristic depletion zone. So it cannot interact with the gel at all. Now it turns out that the receptor's unstructured N-terminal region is critical for this partitioning to occur. So if this N-terminal region is deleted from PEX5 GFP, then the receptor no longer accumulates in the hydrogel. And by contrast, in the absence of the cargo binding TPR domain, the accumulation is unaffected. And specifically, it is the short aromatic motifs, shown here in blue, in the receptor's N-terminal region, which mediate the interaction with the hydrogel. So in the absence of these aromatic motifs, the accumulation of the receptor is also completely precluded. And now we interpret this, these results to mean that these aromatic motifs in the receptor transiently disrupt the cohesive interactions between the tyrosine residues that hold the gel together, and this presumably allows the receptor to melt into the gel and move through it. And we can also reconstitute in vitro the entry of cargo into these YG hydrogels. So in this case, I'm using a solution of non-fluorescent PEX5 and GFP fused to an SKL peroxisome targeting signal. So the SKL, of course, directly binds to PEX5. And in this case, the cargo is also very readily dragged by the receptor into the hydrogel. And this accumulation, of course, depends on the presence of the receptor in the reaction. It depends on the presence of the aromatic motifs in the receptor's N-terminal region. And also it depends on the presence of the peroxisome targeting signal on the cargo itself. In each case, the accumulation is completely abolished. And so based on these in vitro experiments, we originally hypothesized that PEX13 forms a conduit in the peroxisomal membrane that would resemble a very basic nuclear pore. So the conduit is very likely composed of multiple copies of PEX13 and would be filled by a hydrogel-like meshwork within its central cavity or pore. And this meshwork would, of course, function to prevent other proteins from entering peroxisomes, but it would provide an aqueous path for peroxisomal import receptors to selectively move through using the short aromatic motifs in their N-terminal region, and thereby to shuttle a variety of bound cargo across the peroxisomal membrane. We think that the directionality of import would be dictated by the other membrane component of this pathway, PEX14. So PEX14 is also a small membrane protein. It contains a domain that resides on the matrix side of the peroxisomal membrane. And this domain has long been known to bind with nanomolar affinity to the same aromatic motifs in the receptors that we know are important for passing through the PEX13 conduit. And so we think that this interaction with PEX14 would evict the receptors out of the hydrogel on the matrix side of the membrane, and thereby to draw them into the matrix by preventing the back diffusion into the cytosol. So stepping back for a moment, how might PEX13 form a conduit in the first place? So to answer this question, we designed a couple of experiments in vivo using the budding yeast Saccharomyces cerevisiae. So we first asked whether the YG domains of PEX13 really do interact with one another on peroxisomes in vivo, as might be expected if they were assembled into a hydrogel-like meshwork. So to do this, my colleague Yuan Gao introduced cysteines at different positions within the YG domain of endogenous PEX13 in yeast. He then isolated the intact organelles from the corresponding yeast strains and exposed them to an oxidant to see whether disulfide-linked species would be formed. And it's important to note here that the only way to see disulfide-linked forms in this experiment is if the YG domains are close enough in proximity to each other for their cysteines to react. And I'll also point out that PEX13 in yeast contains no other native cysteines. So indeed, when Yuan introduces single cysteines at different positions in the YG domain, he observes very efficient dimerization of PEX13 in the presence of the oxidant. And the formation of these dimers requires an intact membrane because in the presence of detergent, their formation is abolished. And in addition, these dimers are disulfide linked because their formation is also abolished in the presence of the reducing agent DTT. And perhaps more revealingly, when Yuan introduces two cysteines simultaneously into the YG domain of the same PEX13 molecule, he now observes extensive laddering of disulfide linked species. And just by counting the bands in this experiment, we can estimate that at least 10, 12, or 14 PEX13 molecules, and possibly even more, are very likely interact with one another on peroxisomes in vivo at any one time. And this, of course, is consistent with PEX13 forming a highly multivalent meshwork in the membrane. We think that this meshwork is suspended in the membrane by the long amphipathic helix of PEX13, or AH. So the AH is the only other conserved feature in the protein apart from the YG domain, and it has the right properties to line an aqueous channel in a hydrophobic membrane. And very unusually, the AH segment also appears to span the membrane in two possible transmembrane orientations, which we think allows the YG meshwork to be suspended within the plane of the bilayer. Now, Yuan could demonstrate this by introducing different protease cleavage sites at either end of the downstream transmembrane segment in the yeast protein. So the TM segment is, of course, not conserved, but its presence in the yeast protein makes it easy for us to assess the protein's topology in the membrane. So when Yuan exposes the corresponding intact organelles to the orange protease, for example, and then blots for a flag tag, which he engineered at the N-terminus of this chimeric protein, he consistently observes that only half of the PEX13 molecules are ever accessible to the protease. And he sees the same result using the other blue protease here, except now the cleavage product is a bit shorter. And when he adds both proteases together, he can convert the entire PEX13 population on peroxisomes into the two possible cleavage products. And of course, in detergent, each protease has full access to all of the PEX13 molecules, so all of them are cleaved under each condition, as you might expect. And so PEX13 appears to reside in the membrane in two different orientations, such that half of the molecules have the C-terminus in the orange protease site exposed to the cytosol, and the other half of the molecules have the N-terminus in the blue protease site exposed to the cytosol. And it turns out that these two orientations don't correspond to different complexes, but rather they actually interact with each other. And Yuan could show this by retaining one of the protease cleavage sites to mark the two orientations, and also by introducing a cysteine into the YG domain. And so when he cleaves this population on peroxisomes using a protease, he can reveal the two topologies as I've shown you previously. But then when he oxidizes this cleavage reaction, he can convert the two cleavage products into three disulfide link forms. So the first of these, the topmost band, corresponds to a disulfide link dimer between two uncleaved PEX13 molecules. So these would both have the C terminus and the protease cleavage site facing the matrix. The bottommost band corresponds to a combination of two cleaved PEX13 molecules. So two molecules with the protease cleavage site facing the cytosol. And then the middle band corresponds to the combination of one cleaved and one uncleaved PEX13 molecules disulfide bonded to each other, again, as shown in this diagram on the right. And so we can extrapolate from this experiment that the YG domains of PEX13 have an equal probability of interacting with a neighboring subunit in any of the two possible orientations. Now, contrast that to the behavior of cysteines introduced within the amphipathic helix of the protein. So regardless of where UN places cysteines in this AH segment, he always observes the formation of only one disulfide linked form in this experiment. And this corresponds to the combination of one cleaved and one uncleaved PEX13 molecule disulfide bonded to each other. And so this reveals that the AH segments can only interact with a neighboring subunit that is in the opposite transmembrane orientation. And so this can be visualized using the following model, where we've arranged in multiple AH segments around a ring to illustrate how we think they might span the membrane. And so in this putative pore, the neighboring AH segments are, of course, arranged in alternating transmembrane orientations. The corresponding downstream transmembrane segments would also be in opposite orientations. However, the corresponding YG domains, shown here in pink, would all be mixed together within the central channel of this putative structure, where they would be able to form a hydrogel-like meshwork. And this model is actually supported by AlphaFold. So we can use AlphaFold to predict the interaction between 12 or 14 AH segments, which corresponds to the largest oligomeric species we can detect by disulfide cross-linking. And completely agnostically, AlphaFold predicts a very plausible-looking ring-like structure. So in this structure, the neighboring AH segments are arranged in alternating orientations. The height of the structure, it would be compatible with the thickness of a lipid bilayer, so around 4 nanometers. The diameter of around 10 nanometers, it would be consistent with cargoes that are known to be imported into peroxisomes. And interestingly, AlphaFold places the hydrophobic surface of the AH segments around the periphery of the pore, which would be consistent with this residing in the lipid bilayer. And by contrast, the hydrophilic surface is placed facing the central lumen, where we think the hydrogel meshwork would be formed. Now, I'll stress that this model is, of course, speculative, and the actual structure of the import conduit is not yet known. So the nuclear pore-like model that we proposed can explain how folded proteins can be moved into peroxisomes. However, it raises a fundamental conundrum. So if the receptors are supposed to accompany the cargo all the way into the matrix during import, as our model would predict, then how would the receptors return back to the cytosol after import? So to understand how this occurs, a former colleague and I developed a cell-free system that recapitulates a protein import into peroxisomes. So the system is based on a cytoplasmic extract from the eggs of the frog Xenopus laevis. So the extract contains peroxisomes and all other import components. And it allows me to directly monitor protein import by adding fluorescent proteins fused to a peroxisome targeting signal. So the S-scale peptide, for example. And so these reporters would be imported into peroxisomes by the endogenous receptor in the extract, and would then show up as bright puncta in the extract over time, and as shown in this image on the right. And so one major advantage of this cell-free system is that I can very easily manipulate the peroxisomal input receptor. So for example, I can remove the endogenous receptor from the extract by immunodepletion, which of course precludes import from occurring. And I can then fully restore import by adding back the purified recombinant receptor, as shown here on the right. So using this system, I first asked whether the input receptors really do move completely into the matrix during import, as our model would predict. To do this, I devised a strategy that was inspired by earlier work from the lab of Suresh Subramani. So the strategy is based on a unique property of some peroxisomal proteins which do not use the canonical PTS1 import signal. So these proteins use an alternative signal called the PTS2, which resides at their N-terminus in this case. And in higher animals like humans and frogs, this PTS2 signal is normally cleaved off after import by a trypsin-like protease located inside the matrix. And so cleavage by this matrix protease provides a very sensitive assay for entry into peroxisomes. And so I simply took the cleavage sequence for this matrix protease and inserted it at different positions along PEX5, either at the N-terminus or in the middle of the protein or at the C-terminus. And so if PEX5 does fully enter the matrix during import, then we would expect that each of these constructs should be cleaved, and the corresponding cleavage products would accumulate in peroxisomes. And so I replaced the endogenous PEX5 in the extract with each of the cleavable constructs. I allowed them to equilibrate between the cytosol and peroxisomes, and then I isolated the corresponding peroxisomes from the extract by flotation through a density gradient. And I hope you can appreciate that each of the cleavable constructs is quantitatively cleaved during the input reactions, and the corresponding cleavage products are greatly enriched in the peroxisomes compared to the total egg extract. And of course, when the cleavage sequence is scrambled, then none of these constructs is cleaved. So the reaction is specific. We infer from this experiment that PEX5 very likely does move into the matrix during import. But then getting back to our original conundrum, how would it then return to the cytosol? So in the case of nuclear transport receptors normally can move bidirectionally through nuclear pores. However, we think that in the case of peroxisomes, this would not be possible because the interaction with PEX14 inside the matrix should keep the receptors inside the organelle. And so in the case of peroxisomes, a different mechanism appears to be at play. So it has long been known that peroxisomal input receptors have to be ubiquitinated in order to return back to the cytosol. A single monoubiquitin appears to be sufficient for this recycling to occur. And surprisingly, this monoubiquitin is directly conjugated to the conserved cysteine at the receptor's N-terminus. This is, of course, very unusual because typically lysine residues are the preferred site of ubiquitination in proteins. This monoubiquitin is installed by a transmembrane ubiquitin ligase composed of 3 subunits called PEX2, PEX10, and PEX12. And the ubiquitin is thought to recruit a dedicated AAA ATPase formed from PEX1 and PEX6. And this presumably extracts the receptor out of the organelle. Now, the problem here is that ubiquitination can only occur in the cytosol. And so it is not intuitively obvious how PEX5 would be ubiquitinated if it is supposed to be inside the matrix as our model predicts. And so a clue to how this occurs came from identifying a stalled recycling intermediate of PEX5 on peroxisomes. So when the conserved cysteine in PEX5 is mutated to an alanine, the receptor can no longer be ubiquitinated, and thereby it cannot be recycled back to the cytosol. And so you would then expect that the corresponding mutant would only undergo a single round of import and would then become trapped on the organelle. And indeed, the corresponding mutant can only import a very small amount of cargo in the egg extract system, which only form very faint puncta whose brightness does not continue to increase over time, which is quantitated on the right here. This is in contrast to the wild type receptor, which can keep on importing more and more cargo over time. And as a further control, we can also use a PEX5 mutant that lacks the short aromatic motifs in the N-terminal region. These motifs are, again, required for the receptor to move through the PEX13 conduit, and in their absence, the receptor cannot enter peroxisomes and is therefore completely inactive. These phenotypes are reinforced by looking at the accumulation of these different mutants on peroxisomes. So, for example, in the absence of the aromatic motifs, the receptor cannot enter the organelle altogether, and accordingly, the corresponding mutant is undetectable in peroxisomes that had been isolated from the egg extract. Now, by contrast, in the absence of the conserved cysteine, PEX5 can still enter, but it cannot be recycled. And so the corresponding mutant greatly accumulates on peroxisomes compared to the wild type receptor. This, of course, raises the question of where on peroxisomes the cysteine mutant is actually trapped, either inside the matrix or outside the organelle. To make this distinction, we can use protease protection experiments. So if we add a promiscuous protease like proteinase K to the outside of peroxisomes that had accumulated the cysteine mutant of PEX5, we can discern whether the mutant receptor is inside the matrix and therefore protected from the protease, or if it is outside the organelle and therefore fully susceptible to the protease. And what we actually observe is that these mutant receptor molecules are by and large protected from the protease. They only have a small fragment removed from one of their termini. And this shows up as a slightly truncated band compared to the full-length receptor. This is, of course, indicative of the cysteine mutant residing in the membrane in a transmembrane topology. Such that one end of the receptor would be facing the cytosol and accessible to the protease, whereas the remainder of the protein would be on the other side of the membrane and therefore protected from the protease. And I've drawn the receptor in this orientation here, again, for simplicity, but it turns out that this is the actual conformation of the protein in the membrane. And I can demonstrate that by adding a triple flag tag to either the N-terminus or the C-terminus of this mutant receptor. And so if the orientation is correct, as I've drawn it here, then you would expect that in the presence of proteinase K, a larger chunk of the N-terminal tag construct should be removed compared to the C-terminal tag construct, and also that this N-terminal construct should lose all flag immunoreactivity. And that's exactly what I see. So with the N-terminal flag tag construct, the receptor produces a much smaller truncated form in these experiments compared to the C-terminal tag construct. And the smaller truncated form is no longer immunoreactive for the flag tag. So we interpret these experiments to mean that PEX5 does move completely into the matrix during import, together with the bound cargo. and that it then reemerges such that its N-terminus would face the cytosol and allow the conserved cysteine at the very N-terminus to have access to the ubiquitination machinery in the cytosol. Now, we don't think that the receptor is moving directly through the lipid bilayer, as this cartoon naively suggests. Instead, PEX5 appears to contain an export signal out of peroxisomes. The signal consists of a short amphipathic helix, or APH, that we discovered within the N-terminal unstructured region of the receptor. So this APH is characterized by conserved hydrophobic amino acids, shown here in yellow. And these hydrophobic amino acids, when mutated, and the corresponding APH mutant is inactive in the egg extract system. And to be more precise, the actual phenotype of the APH mutant exactly mimics the phenotype of the cysteine mutant of PEX5. So if you remember, the cysteine mutant can still enter peroxisomes, but it cannot return to the cytosol. And so it can only undergo a single round of import and forms puncta whose brightness does not continue to increase over time. And so this phenotype suggested that the APH very likely also plays a role in receptor recycling. And this hypothesis is, of course, supported by protease protection experiments. So in the absence of the APH, PEX5 accumulates on peroxisomes to a similar extent as PEX5 lacking the conserved cysteine. Now, in the case of the cysteine mutant, the receptor very clearly stalls in a transmembrane orientation that can be revealed as a slightly truncated form using protease protection experiments. However, in the absence of the APH, PEX5 is now completely protected from protease digestion in these experiments. And this, of course, suggests that in the absence of the APH, PEX5 is now trapped completely inside the peroxisomal matrix and doesn't even cross the membrane. To understand why this would occur, I prepared peroxisomes in the egg extract that had accumulated either the cysteine mutant of PEX5 or the APH mutant. I then purified the corresponding receptors using a built-in flag tag and identified any co-purifying proteins by mass spectrometry. And so the results are presented in terms of the number of peptides detected for the components on the right here in two independent experiments. And so in the absence of the conserved cysteine, we can pull down both components of the import conduit, PEX13 and PEX14. We can also detect all three subunits of the ubiquitin ligase complex that ubiquitinate the receptors during recycling. In the absence of the flag tag, of course, none of these components is pulled down. But in the absence of the APH, the import conduit components are still efficiently isolated, but now all three subunits of the ligase complex have been completely lost and are undetectable. And so we interpret this to mean that the ubiquitin ligase complex very likely moonlights as a retrotranslocon that would allow the receptor to reemerge in the cytosol. And so we can understand how this would work by looking at the cryo-EM structure of the ubiquitin ligase complex that had been solved by my colleague, Pei-Chung Feng. So in the structure, all three subunits of the complex co-assemble into a funnel-shaped bundle in the membrane with an open pore spanning the bilayer. And this pore here in the middle provides a very clear aqueous path between the matrix and the cytosol, through which we think the receptor's N-terminus would reemerge, as shown here on the right. All three of the ligase subunits contain ring finger domains that are required for the ubiquitin ligase activity. In the structure, all three ring domains pack together into a tower on the cytosolic side of the membrane, right above the open pore. And this configuration would be ideal to ubiquitinate the receptor's N-terminus as it reemerges across the membrane. This ubiquitination would presumably then recruit the PEX1, PEX6 ATPase, which could then pull the receptor out of the membrane and return it to the cytosol. Now, the problem with this model, again, is that the receptor also contains a large and well-folded domain at its C-terminus that binds to cargo. This is the TPR domain. However, the pore in the ligase complex is only 1 nanometer wide, so much too small to accommodate such a large folded domain, which is shown here roughly to scale, right? It's only large enough to accommodate a single extended polypeptide. And so we think that in order for the entire receptor to be pulled across the membrane in this way, the cargo binding domain would have to be unfolded, presumably by pulling by the PEX1, PEX6 ATPase on the opposite side of the membrane. And we can test this hypothesis using the xenoplus egg extract system in the following way. So normally, a wild-type PEX5 can only import cargo that contains a peroxisome targeting signal. So for example, GFP fused to the SKL peptide. Plain GFP without a targeting signal, of course, cannot be imported because it cannot bind to the wild-type receptor. Now, if PEX5 were fused to a GFP binding nanobody, or NB here shown in green, then both GFP-SKL and plain GFP should bind to this chimeric receptor. And in this case, GFP-SKL should still accumulate in peroxisomes regardless. But the only way for GFP to accumulate would be if the chimeric receptor were unfolded during each import cycle in order to break the interaction between the GFP and the nanobody and leave it behind inside the matrix. And by contrast, if the receptor were not unfolded, then the GFP would remain bound and it should not accumulate. And what we actually observe is the following. So with the wild-type receptor, only GFP-SKL can accumulate inside peroxisomes as expected. Plain GFP cannot. And this, of course, corresponds to multiple rounds of import because the corresponding cysteine mutant shows only very faint puncta by comparison. And then in the case of the chimeric receptor, both GFP-SKL and GFP alone can be very efficiently imported into the peroxisomes in the extract. And again, the puncta that you're seeing here correspond to multiple rounds of import because the corresponding cysteine mutants do not show comparable accumulation. So we can put everything together to derive the following model for how we think proteins would be imported into peroxisomes. So peroxisomal proteins would first be recognized in the cytosol by import receptors like PEX5. The receptors would then shuttle the bound cargo across the peroxisomal membrane using a conduit formed by PEX13. And this conduit would, of course, be filled by a hydrogel-like meshwork that would selectively allow import receptors to move through, but would exclude other proteins. The directionality, of course, would be dictated by PEX14 that would prevent back diffusion of the receptor into the cytosol. And so after moving into the matrix, the receptor's N-terminus would insert into the open pore in the ubiquitin ligase complex and thereby reemerge in the cytosol with the conserved cysteine being exposed for ubiquitination. This would, of course, ubiquitinate the receptor on the conserved cysteine. The ubiquitin would then recruit the PEX1, PEX6 ATPase. The ATPase would, of course, pull the receptor out of the organelle through the membrane. And this pulling, we think, very likely would unfold the cargo binding domain, which would strip off the cargo and leave it behind inside the matrix. And this, of course, provides a very simple mechanism for how cargo might be released. And finally, the cargo binding domain presumably would refold in the cytosol, and the ubiquitin would be removed by deubiquitinases in the cytosol, which would reset the receptor for another round of import. And I'll just close by pointing out that this whole cycle is very likely driven by the export of receptor molecules out of the organelle. And so as long as the receptor can be efficiently recycled, then cargo proteins can be moved into the organelle against the concentration gradient. And we hope that this model will now provide a comprehensive framework for understanding how mutations in these different input components might cause disease, and perhaps in the long term, this would reveal possible therapeutic strategies. And so with that, I would like to thank my advisor, Tom Rappaport, for his support over the past few years, and my colleagues Pei Chun and Yuan, also the Imaging Center at Harvard Medical School for microscopy assistance, and perhaps most importantly, the Helen Hay Whitney Foundation and HHMI for their very generous support of this project over the past few years. And with that, I'll take any questions, and thank you very much.
Well, thank you, Mike, for this beautiful talk. So yeah, as Yihang said, please put your questions in the chat, and I can read it to Mike, or Mike, you can read them yourself, of course. So we have one question already, which is Liang Gi asks, what's inside the peroxisome which unfolds PEX5?
Right. So we don't think PEX5 is being unfolded inside the peroxisome. We think it is the PEX1, PEX6 ATPase, which is pulling, it's tugging on the receptor from the cytosolic side. And we think that this pulling would destabilize the cargo binding domain on the matrix side of the membrane. Like imagine if you had a ball of yarn and you were pulling on one strand, that would presumably unfold the remainder of the ball of yarn. At least that's the idea.
So the next question is from Shenji Wu, who asks, does the length of the FG repeats affect the hydrogel formation?
Right, so that is something we haven't yet explored. One interesting detail is that the YG domain of PEX13 is often quite a bit shorter than FG nucleoporins. The domain is also quite variable between different organisms. And so obviously this works in vivo, and we've tried to swap different YG domains from different species to see if they could replace the yeast YG domain. And they more or less are functionally interchangeable. So we don't think the length necessarily matters, but what role it plays, we haven't yet explored extensively.
Okay, so the next question is from Yihang, who asks, how efficiently can PEX refold in the cytoplasm? Can PEX5 refold in the cytoplasm? And do you think that this happens spontaneously or with the help of some factors there?
Right, so that is still a hypothesis. We haven't yet looked to see if the cargo binding domain could spontaneously refold. Presumably it can, because it's composed of, again, repeating TPR repeats. But we can't discount the role for cytosolic chaperones in this process as well. I think this is something that could be worked out in the future.
Okay, so next, Yusong Guo asks, what provides the force to push PEX5 into the peroxisome?
Right, so that's also an interesting question. So we obviously don't know yet what allows the full receptor molecule, and more importantly, the bound cargo, to also move through the PEX13 conduit. One possibility is that the receptor's N-terminus can initiate recycling before the whole molecule has moved into the matrix. So I'll just point out that all of the features required to recycle the receptor are located near the N-terminus. Right? So you can imagine that the N-terminus could insert into the ubiquitin ligase before the remainder of the molecule had moved across the membrane. And perhaps this would allow the PEX1, PEX6 ATPase to pull the remainder of the molecule across the pore, sort of on the back side of the pathway.
Okay, so the next question asks, what happens to cargo in the matrix? Does PEX5 function as an importer or modify the cargo that helps the cargo during degradation, for degradation?
So the cargo, in the case of peroxisomes, corresponds to all of its metabolic enzymes and other proteins it needs to perform its different functions. So as far as we know, PEX5 and this whole import pathway predominantly functions to deliver the metabolic enzymes into the organelle. I'll point out that, interestingly, the ubiquitin ligase complex uncannily resembles ubiquitin ligases involved during ER-associated degradation, or ERAD. And so it's possible that the ubiquitin ligase complex might have some role in retrotranslocking cargo proteins that are unfolded or damaged by analogy to ERAD, for example. But again, this hasn't been explored systematically.
Okay, so the next question asks, what might be the driving force to bring to concentrate proteins into the matrix of the the questioner asks, I understand that PEX14 could serve as an anchor once it gets in.
Right. So we think the force here is mostly thermodynamic. So it seems to be the concentration gradient of the import receptor itself, sort of across the peroxisomal membrane, that appears to be driving the whole pathway. And this gradient is maintained sort of, or is very steep, because the receptor is continually recycled out of the matrix. And then PEX14 just provides an avidity sink on the matrix side, which we think would facilitate the attraction of the receptor into the organelle, if that answers the question.
Okay, so the next question is, if you survey the genome, are there other YG repeat containing membrane proteins that may indicate that this mechanism may also work for other organelles?
So I think that is a very interesting question. It's unclear whether other organelles have similar proteins, but there are similar proteins in the genome that utilize tyrosines to phase separate. A well-known example comes from the work of Steve McKnight, who showed that the protein FUS, or fusin sarcoma, which is a major component of stress granules, also relies on unstructured regions rich in tyrosines to phase separate in the cytosol.
So next, someone asks, any speculation about the association, close relationship of the endoplasmic reticulum and peroxisomes in mammalian cells during normal and disease states, as with mitochondria?
So is the question about the relevance of the
Yeah, I think that's what the person is asking here. But
right.
Yeah.
I think whether that plays any role in protein import is unclear, but it's certainly very attractive that the juxtaposition between peroxisomes and other organelles would facilitate the transfer of metabolites and also lipids, which Will's previous work has highlighted.
So the next question is, will the hydrogel keep ions from exchanging?
Right. So this has been a topic of some contention in the field. It seems that peroxisomes do not maintain steep gradients of ions or protons across their membrane. They also appear to be permeable to many small molecules, so molecules under a few hundred daltons in size. So both the PEX13 conduit and the ubiquitin ligase complex are, of course, permeable to small molecules, right? The hydrogel is an aqueous meshwork, and the ligase complex contains an open pore, which has no obvious plug based on our structure. So this could explain in part why peroxisomes are not known to maintain ion gradients across the membrane. Yes.
So I guess I'll ask the last question, which is more a speculative question. How do you think this complex pore, I mean, the structural prediction you showed was really beautiful, but how do you think that kind of structure gets assembled? And does it need helpers to do that? How would that work, do you imagine?
Right. So we scratch our heads about that every day in the lab. So proteins with a dual topology like PEX13 are extremely unusual in life in general. We're not sure exactly how the pore would be assembled. It's possible, or it would be unlikely that PEX13 would span the membrane as monomers, because then the hydrophilic surface of the AH segments would contact the lipids in the membrane, which should be unfavorable. So we favor the model where PEX13, I don't know, might initially be in one orientation or might be lying peripherally on the membrane. And then once it reaches a certain threshold concentration, perhaps the assembly is just a concerted all or none event, and the protein spontaneously assembles into a favorable structure of the type that we hypothesize. But of course, that remains speculative. I mean, there might be membrane chaperones, but it also depends on where PEX13 would be inserted into the membrane, either the ER or perhaps it's directly inserted into the peroxisomal membrane, but we don't know yet.
All right, so we do have one more question. So the questioner says, elegant and beautiful work. A prior question relates to mine, asking if there are any chaperones present inside or out, say like trap 1 of mitochondria, a role in oxidative stress in cells, neurodegenerative diseases, etc. Thank you for all. Looking forward to your future findings.
So in terms of chaperones in peroxisomes, there are not many that are known. I think the only one that has been, at least in part, confirmed is a LON-type protease that is known to reside in the matrix. It has an ATPase domain that some have argued could have chaperone activity. Whether that plays any role in protein import, I think hasn't been explored.
Okay, well, seeing no more questions, I'd just like to thank Mick again for this fantastic talk and for you all for attending and for these great questions. And thank you again.
Yeah, thank you very much for having me.
Thank you, everyone. Bye-bye.
All right. Bye-bye.
Thank you.